Welcome to today's Explainer. We're going to dive right into an absolutely epic 13-year journey. We are talking about a massive quest that officially kicked off in 1990 and didn't wrap up until 2003. It was a mission to map the absolute most fundamental, unexplored territory in existence, the human body.
So I want you to ask yourself a question. What would it actually take to read the literal instruction manual for a human being? I mean, if we think of our biological genome as a tangible book of life, how do we even begin to, open the cover, let alone read the text inside?
To answer that, we first have to wrap our heads around the mind-boggling scale of what we're actually dealing with here. The human genome consists of approximately 3 billion chemical base pairs. 3 billion. Think about that for a second. That's 3 billion individual rungs on the DNA ladder that dictate the whole shebang, everything from your eye color to your susceptibility to certain diseases. Now, what's truly staggering is the sheer cost of tackling those 3 billion base pairs. Back when this project was first launched, the estimated cost of sequencing was about $3 per base pair. So, do the math, and you're looking at a monumental $9 billion mega project literally just to get the very first read-through. Insane, right?
If dollars and base pairs are feeling a bit abstract, let's try visualizing it like this. If you were to take that genetic sequence and physically type it out into books, it would fill 3,300 of them. And we're not talking about small paperbacks here. Every single one of those 3,300 books would have 1,000 pages. That is the invisible scale of human biology made visible.
Section 1. Humanity's biggest megaproject.
Because of this massive, multi-billion-dollar scale, the Human Genome Project, or HGP, wasn't just some regular science experiment. It was a massive global undertaking. Let's look at the actual goals they set out to achieve. First off, they needed to identify all the estimated 20,000 to 25,000 genes in human DNA. They had to sequence those 3 billion base pairs we just talked about, but it wasn't just about finding the data, right? They had to store this colossal amount of information in databases and transfer the related technologies to other sectors, like industry. And crucially, they also sought to address the ethical, legal, and social issues that such profound knowledge would inevitably bring up. This was actually a huge, groundbreaking move for a biology project, recognizing early on that reading our biological source code carries profound societal weight. The demands of storing and analyzing all this data were literally so intense that it forced the rapid development of an entirely new field in biology called bioinformatics.
Section 2. Methods of decoding DNA.
So the crucial point is, how did they actually do it? How do you read a biological text of this magnitude? They basically had two major approaches. On one side, we have expressed sequence tags, or ESTs. This is kind of like skimming the manual to only read the action verbs. They focused solely on identifying the genes that are actually expressed as RNA. On the other side, you've got sequence annotation. Now this is the blind brute force approach. You sequence the entire genome, both the coding and non-coding parts, cover to cover, and then you just try to figure out what the different regions actually do later on.
To read it cover to cover, they needed biological machinery to basically copy the manual. Enter BAC and YAC. That's bacterial artificial chromosomes and yeast artificial chromosomes. You can think of these specialized vectors as giant cellular printing presses. Because DNA is such a long polymer, there are strict technical limits to sequencing really long pieces all at once. So scientists use bacteria and yeast as hosts to clone and amplify smaller fragments of DNA. That way, they could actually be sequenced with ease.
And this brings us to the sequencing pipeline itself. It's a beautifully logical step-by-step process. First, you isolate the total DNA from a cell. Second, you break it into smaller random fragments. Third, you clone and amplify those pieces in our BAC or YAC hosts. Fourth, you sequence them using automated DNA sequencers based on the famous Sanger method. Now, the Sanger method was the absolute gold standard of the era, but scaling it up to handle 3 billion base pairs, that was a colossal technological hurdle they had to overcome. And finally, step 5, align the overlapping fragments. This alignment was humanly impossible, by the way. I mean, nobody's doing that by hand. So specialized computer programs were developed to digitally stitch the whole genome back together.
Section 3. Surprising genomic features.
Once this massive computational pipeline produced the first readable draft, well, scientists were in for some major plot twists. Here's one of the absolutely biggest surprises. Before this project, scientists estimated humans had anywhere from 80,000 to 140,000 genes. But the actual total number? It's estimated at just around 30,000.
Honestly, it was a bit of a blow to the human ego to realize we operate on far fewer unique genetic instructions than a lot of much simpler organisms out there.
And that brings us to another wild discovery. Less than 2% of our genome actually codes for proteins. The vast majority, over 98%, consists of non-coding, repeated sequences. These are basically stretches of DNA repeated hundreds or thousands of times. And while they don't code for proteins, they are shedding incredible light on chromosome structure, dynamics, and human evolution. So it turns out, what some folks previously just dismissed as genetic filler, yeah, it's actually vitally important.
We also learned how wildly varied our genetic instructions are in size. Your average human gene consists of about 3,000 bases. But then you have these extreme outliers, like the gene for dystrophin. Dystrophin is the largest known human gene, measuring in at a massive 2.4 million bases all by itself. Just incredible.
Information density is also incredibly imbalanced across our genome. Chromosome 1 is packed to the brim with the most genes, 2,968 of them to be exact. On the flip side, the Y chromosome has the absolute fewest, with just 231 genes. So our instruction manual is definitely not formatted evenly.
Section 4. The S&P Revolution.
Now we're getting to the real climax of this explainer, the part that fundamentally changes how we view ourselves and our health. The tiny variations that make every human unique. Despite all our visible differences in height, hair color, and facial features, here is a profoundly beautiful realization from the source material. Almost all nucleotide bases, 99.9% of them, are exactly the same in all humans. You and I, we're 99.9% genetically identical.
So what exactly makes up that 0.1% difference? Well, scientists identified about 1.4 million locations where single-base DNA differences occur. These are called single nucleotide polymorphisms, usually just pronounced as SNPs. You can think of them as 1.4 million highly specific typos in our manual. And these SNPs are incredibly crucial. They hold the promise of revolutionizing how we find chromosomal locations for disease-associated sequences and even how we trace the history of humanity itself.
Section 5. The Future of Biology.
By completing this map, the Human Genome Project fundamentally shifted the future of medical and biological research. In the past, researchers could really only study one or a few genes at a time. Now, because of this mega project, they have a radically new approach. As the source material beautifully puts it, researchers can now study how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. It's a huge transition. We went from looking at isolated puzzle pieces to finally seeing the holistic, entire picture of human biology.
Which leaves us with this final thought. We've successfully mapped the human genome as a species, but every single one of us carries our very own unique version of this 3 billion-letter manual. So, what secrets is your own personal genetic code still hiding, just waiting to be uncovered?